Hi, I'm Katie, and we're going to talk to you today about renin.
I'm Nora. Are there times when you ever feel so mad that you just feel like breaking things or throwing something? Well, at times like this, your blood pressure is most likely through the roof. You can blame renin for this.
According to a 2002 article published in the Journal of Clinical Investigation, renin is a hormone which catalyzes the first step in a series of reactions which regulate arterial blood pressure.
Renin is both considered a hormone and an enzyme, but today we will be focusing on the enzyme portion of renin.
Renin is a highly specific enzyme that plays a major role in multiple physiological systems throughout the body.
First, we will be discussing the basic bioinformatic information about renin, and then we will examine predictions about renin, and finally we will discuss practical applications of the enzyme.
First, let's examine a few biochemical characterizations of renin.
Renin was first sequenced in 1988, and it is 406 subunits long. You might also recognize renin by its other names, angiotensinogenase or REN. According to a UniProt search, renin's enzyme code is 3.4.23.15, and its molecular weight is about 45,057 daltons. Renin can form two sequences or isoforms, and those can be processed by using alternative splicing, but its most common isoform is shown here in this long 406 subunit sequence.
Renin is naturally found in Homo sapiens, and other primate relatives have sequences which are 90 to 99% similar. The gene responsible for renin production is located on chromosome 1, shown here, and is primarily expressed by granular cells in the kidney. The gene product is pre-prorenin, and it contains a signal sequence which directs ER targeting, glycosylation, and proteolytic processing, resulting in prorenin and renin production. No non-lethal mutations of the renin gene have been reported to date, with the exception of premature stop codon mutation, which leads to benign hyperprorenemia. However, recessive mutations causing complete loss of renin synthesis, resulting in renin tubular degenesis, have been described.
The primary structure of renin contains double domains. The amino and carboxyl termini contain areas of similar sequence, but these double domains form a two-lobed structure surrounding the active site. Renin's secondary structure consists of alpha helices and beta strands, but the structure consists mainly of beta strands. And so in this figure, the alpha helices are characterized by the blue sections, the beta strands are the pink sections, and the beta strands are the green sections. The catalytic region contains two critical aspartic acid residues. One is contributed by each half of the molecule. And in contrast to other proteases similar to renin, renin is highly specific for its substrate, angiotensinogen, and it's most active at a neutral pH. Human renin is reported to be a glycoprotein. Aspartic acid residues are important to the enzymatic activity of renin and are located on a separate domain as two glycosylation sites, which are also on the enzyme. The two acidic groups which are directly involved in the catalytic activity of renin are aspartates located at position 38 and 223. It is likely that both of these contribute to the structure of the active site due to the fact that they are both located in the middle of their separated exons.
Renin's function is to create angiotensin 1 from angiotensinogen. This is the first step in a series of reactions which leads to elevated blood pressure and increased sodium retention. It does this by hydrolyzing a single peptide bond in angiotensinogen to produce the decapeptide angiotensinogen 1. Renin differs from other proteases, which can act on a broad range of substrates in that it is highly specific. The Renin-Angiotensin system has also been found to have widespread and diverse roles, including modulating vascular tone, renal sodium handling, erythropoiesis, thirst, cardiac hypertrophy, and functioning through the local renin-angiotensin systems in many organs. The renin-angiotensin system is one of the major regulators in blood pressure and fluid and electrolyte homeostasis. Primary components of the renin-angiotensin system are angiotensin, renin, and the angiotensin converting enzyme, angiotensin 2, angiotensin 2 receptor, and the binding of angiotensin 2's receptor mediates vasoconstriction and aldosterone and catecholamine release, as well as drinking, secretion of prolactin and adrenocorticotropic hormone, and glycogenolysis.
Angiotensin is a globular glycoprotein, which is the major substrate for renin and the precursors of angiotensin 2. It shares homology with A. antitrypsin and belongs to the superfamily of serine protease inhibitors, or SERPINs. Angiotensin 1 is angiotensin 1 is cleaved by renin at a leucine-valine bond in the amino-terminal region of human angiotensins and of other species. Because of its large size, angiotensinogen has been hypothesized to have other functions. And evidence for this is that angiotensinogen levels are increased during inflammation. Analysis of the human genome indicates that there is a single gene for angiotensinogen, and it's comprised of 5 exons and 4 introns. And a recent study has linked certain variations of this gene to inherited essential hypertension or high blood pressure. Physiological studies of alterations in plasma levels of angiotensinogen have provided evidence for upregulation of this protein during pregnancy or estrogen therapy, and in Cushing's syndrome and downregulation of it after thyroidectomy and in Addison's disease. The major source of angiotensinogen is in the liver, and this has been confirmed by measurement of the distribution of angiotensinogen mRNA in various tissues.
Renin is produced in the juxtaglomerular cells of the kidney cortex in very small amounts. Because of this, detailed biochemical analysis has been limited. Renin, which has been purified from human tissues, has shown variation in molecular weight and amino acid composition. It has been experimentally determined to be approximately 36,000 Daltons and 337 amino acids long. Recent evidence using molecular and biochemical approaches to angiotensin physiology raises the possibility that there are distinct local renin-angiotensin systems with different mechanisms of regulation. This is different from the theory that there exists one systemic hormonal system through which various components are derived from different organs and then delivered to their site of action by the circulatory system. Local renin systems have been proposed in the vasculature, brain, heart, and kidney. However, there is not enough evidence for the existence of all the necessary components of the systems in physiologically relevant amounts.
Okay. Some practical applications of renin. Restriction mapping and linkage analysis have been used to study the association of renin with specific diseases such as hypertension or high blood pressure. Linkage analysis uses a mathematical model to determine whether a particular phenotype and a genetic marker co-segregate. There has been interest in developing renin inhibitors as a therapeutic approach to hypertension based on the idea that renin is a highly specific enzyme and is the primary step in this cascade. Though several classes of renin inhibitors have been developed, more are still undergoing evaluation for therapeutic efficacy and have not yet reached the market for antihypertensives.
Now we're going to talk about practical applications of renin. Authors Li, Wei, Chen, Liu, and Cao, and their paper 1,25-dihydroxyvitamin D3 is a negative endocrine regulator of the renin-angiotensin system, was first published in 2002.
In this paper, they hypothesized that vitamin D was a negative regulator of renin expression in vivo or in living organisms. And in order to test this hypothesis, they had mice as their treatment subject, and they had a couple different treatment groups. The wild type mice were their control group, and VDR null mice were one of their treatment groups. And VDR null means that they were void, they didn't have a vitamin D receptor. And these VDR null mice were placed on a strict diet in order to normalize their blood ionized calcium levels. And then their sodium intake was increased, and their diet was supplemented with 8% NaCl, and then the wild type mice were placed on a special diet containing strontium until they showed levels of hypocalcemia. And this action was taken in order to examine the effects of blocking vitamin D synthesis.
The results of the study show that VDR null mice have renin levels which are increased threefold compared to the control group. This shows that renin expression is increased in the mutant mice due to disruption in the vitamin D signaling pathway. As a result of this experiment, vitamin D deficient mice were hypertensive, meaning they had high blood pressure because of elevated renin activity. Mice that were given strontium to block vitamin D synthesis became hypocalcemic. Vitamin D is needed to regulate the calcium levels in the blood. As the authors expected, the treatment mice showed increased levels of renin activity. The finding supports their hypothesis that vitamin D is a negative regulator of renin expression. However, elevated renin expression due to suppressed vitamin D levels, not hypocalcemia.
And so overall, their study concluded that vitamin D is a direct suppressor of renin. So next time you think about throwing something or feel your blood pressure getting really high, that's due to renin. So our works cited and all of our sources can be found in the description below, and thanks for watching.